I'm super excited to be here to take you through what I think is going to transform immuno-oncology. We're the first company to develop engineered monocytes and deliver them to patients. We're the first company to engineer in vivo RNA and deliver that to patients as well. And today, I'm going to show you a little bit of that information that we've generated. Just a high level, Myeloid Therapeutics was founded back in March of 2019. Sid Mukherjee and Ron Vale co-founded the company with myself and Tom Cahill at New Path Venture Partners. What we do is we take RNA and immunology and we essentially push these together and have built out a number of proprietary innate immune CARs, and then we deliver them using RNA. And through this process, as you can probably appreciate, coming before the mRNA revolution with COVID, we actually figured out some pretty cool ways to use RNA to deliver genes using retrotransposons, which I'll touch on very briefly at the end, but I think it's interesting for the focus of this meeting. In the context of moving forward, I'm a very aggressive drug developer. I believe that solving human disease can only be done in humans. And so what does that mean? From the first day we did our first animal experiment using our in vivo CAR, it was about 16 months to the first patient was dosed. For our cell therapy, it was about 18 months. And the reason for this is twofold. One, using RNA allows us to move quickly. Secondly, we've built the company with a foundational set of investors that also have the same belief in that we need to iterate, we need to make each product 10 times better. And I'll show you how we've been able to do that during this presentation. I think the most important thing is to point out here is that we have two clinical programs. One is a cell therapy in T-cell lymphoma, and the other one is our in vivo CAR in TROP2 expressing epithelial tumors. In the context of manufacturing, we are trying to be wise about that. We've done a deal with a state in Australia, the New South Wales government, and we're in the process of building out our GMP facility there. And then in the context of fundraising, we've raised around $125 million in equity and have done a number of deals. But why are we here? We're here because I have been through the immuno-oncology landscape. I remember back in 1999 when immuno-oncology was thought to be quite a pathetic field. Obviously that has changed in the last 20 or so years. What hasn't changed though is our focus on T cells. And as an immunologist, the very first thing you get taught, and I taught this as an academic at Northwestern, is myeloid cells, the primary orchestrator of immune responses. If you want a potent T cell response, you need to have good antigen presentation from myeloid cells. They have to provide co-stimulation. Obviously, success in CAR T cells came when Carl and Bruce combined CD3 zeta with CD28, essentially providing those two signals. And so what Sid and Ron and I did is we said, look, no one has been focused on taking all the great technology being applied to CAR T cells, cell and gene editing, engineering, and looking at how this can be applied to myeloid cells. And so that's what we did. Within the myeloid cell compartment, there are a number of pattern recognition receptor pathways you've probably heard of, STING, NLRP3, FC receptors, and so on and so forth. And these pathways are designed to recognize and develop innate and adaptive immune responses during pathogenic challenge. What's really interesting within the context of biology is these same cells that essentially respond to drive immune response are exactly the same cells tumors recruit into the tumor microenvironment. The big difference being once inside the tumor microenvironment, the micro environment, the milieu around it, obviously turns those cells into being somewhat useless. But they have all of the machinery needed to function. And I think if you look at the world we are in now, Moderna and so on and so forth have shown that you can actually harness these cells in vivo to stimulate adaptive immune responses in an adjuvant setting, for example, in melanoma, and also actually in PDAC. So we're really focused on how do we actually get these cells to do what they're designed to do, which is elicit robust immune responses. And as I pointed out, we use a number of ways to do this. First and foremost is we've been using RNA, and I'll touch base very briefly on that. Secondly is we interrogated these pattern recognition receptor pathways. I have a very talented set of protein scientists that have been able to laser lock on the 10 to 12 amino acid sequences from these receptors that drive activity. And so if you think of that in the context of a construct, you can actually build constructs with numerous of these different signaling pathways to drive activity. And so we're very excited by this. And obviously when you have this ability, it just comes down to how do you want to apply those receptors. And so our first approach was using an ex vivo cell therapy. And so what we did here is we took monocytes from the blood. So monocytes in the blood express high levels of a chemokine receptor called CCR2. This is how the tumor attracts them. Now, that exact same monocyte, if it was to go into the COVID lung, would drive inflammation, right? It all comes down to the microenvironment in which it goes into. So what we did is we took those cells out of the apheresis product. We engineered them with RNA. It's a one-day process, delivered back to the patient in 8 days successfully. What you can see here is we have successfully administered this to 3 patients to date. And so what we found in this particular subset of patients is we didn't see any infusion reactions at all. All patients are actually more than 3 times the median overall survival. There are some complicating factors within that that I'm happy to discuss after the meeting. But what gets us very excited is the biomarker data. So this is a baseline biopsy from a patient that had 96% of their bone marrow cancer. 96%. This patient wasn't expected to live 2 months. You can see 17 days after treatment on the right, a reduction in the green, which is T cells, the T cell lymphoma. We see an increase in inflammatory cells, CD14 monocytes, and a decrease, we think, in some of the other TME-associated factors within the macrophage compartment. What else was very interesting is if we look in the periphery of these patients, this is in this particular one, we saw the emergence of 8 new TCRs. So within 17 days, the entire tumor microenvironment was changed. We're seeing the evidence of at least retrafficking, if not emergence of more TCRs. And we actually have biopsy data now showing within the biopsy itself, not just in the periphery, we see these T cells expanding. So the whole idea of being able to engineer these cells, get them to the tumor microenvironment, get them to change the tumor microenvironment, really is playing out in the cell therapy context. And we need to collect more data to obviously really push that forward. But this really pushed us, our thinking along to see how can we do this directly in vivo. And I have experience developing nanotechnology, and one of the things that's really interesting about myeloid cells is their job is to take up anything you put in the blood like a lipid nanoparticle. So the idea that we need to go make a myeloid-specific LNP like some of these other companies are trying to do for T cells, we simply don't have to go and do. So what we said, how do we solve for the fact that the liver and the spleen still are pretty good at taking up non-targeted LNPs? And even for targeted LNPs, they're a formidable barrier. We said, why don't we use protein biology? It's a very simple approach that we did. So thinking of this just pragmatically, on the left here you can see a cell surface receptor made up of 2 subunits. Many immune receptors, as you're aware, are made up of many subunits. The TCR is 8, for example. But what actually happens for these to be expressed is these 2 subunits have to be expressed inside the cell, and they usually come together in the endoplasmic reticulum and then get shuttled to the surface. If you express A in a cell that doesn't have B, you won't get a receptor and vice versa. So what we said is, why don't we just take half of the receptor and fuse a tumor recognition domain to that, express that as the RNA and deliver that as the LNP. And obviously the outcome is only cells that have both subunits are going to be able to express a functional receptor, and cells like hepatocytes that will not be able to. And so this really shows it here. The idea of the therapeutic product is your RNA delivering, half of the receptor, if you like, the CAR, it's infused, and we've actually developed a product that is using the FC alpha receptor, which is a myeloid-specific receptor, and you can see here is made of 2 subunits. The first subunit is the FC common gamma chain. It has 4 ITAMs in it, and the second subunit is CD89. Now you'll notice there is no signaling domains for CD89. What we actually did is we took CD89 and we fused the tumor recognition domain to that, and that is actually what we deliver. And only the myeloid cells are able to express the receptor, and cells that take it up, that don't express the FC common gamma chain, don't. So as the CEO of this company, the biggest challenge in the last 4 years for me has been to convince the investor community that targeting the myeloid compartment can have the same outcomes as CAR T cells have had in hematological malignancy. And apart from proving this in humans, the other step back from there was to take animal models that we know CAR T cells don't work in. And so in this particular model here, this is the B16 over melanoma model. CAR T cells targeting GP75, a protein expressed in this model, do not work. Even if you give them at day 0 copious numbers. Also, checkpoint inhibition doesn't have any monotherapeutic effect either. So what we actually did here is we let these tumours progress to around 75 to 100 millimetres cubed, and then we initiated treatment with the in vivo product, specifically targeting GP75. And what's interesting, apart from obviously the compelling anti-tumour activity, is that when we looked in the tumour microenvironment, only the myeloid cells were expressing the CAR. Now, obviously the whole point of this technology is to have direct myeloid anti-tumour activity, but then to elicit potent T-cell activity. What we found in the periphery of these animals is significant APC activation. We saw a significant reduction in exhausted T-cells in the TME itself. And then what's really exciting is T-cells that were coming into the tumour were activated, they had recently proliferated, and they expressed granzyme B associated with activity. Now, it's all well and good. Many people have made a dent in mouse tumours. Really, our goal here, like I mentioned at the beginning of my presentation, is to figure out how we cure cancer in human beings. And so the very first thing we did is, what is the best place to start taking our technology? And if you've been following the ADC world, you appreciate that TROP2 is a hot target right now within that space. And so that's where we went into. You can see this is a xenograft model. Again, we established the tumour and then initiated treatment here, and we saw significant anti-tumour activity. When we fished out the tumours, the myeloid cells expressed it. This is an important model because there is no T cells here. So this is just looking at the direct anti-tumour activity of the ARM cells themselves. So it is a multifaceted effector function. It is direct anti-tumour activity and other things that I'll show you in a moment to summarise it. Obviously, we had to move into non-human primates where we confirmed not only safety, but also the cell-selective expression of the CAR. And so we're very excited by this. I'm going to move through this for time. I think the most important point here is that we're trying to build a technology that can essentially overcome the need to know what the neoantigens are and the ability to get into the TME, activate it, liberate novel neoantigens. At least in the animal models, we know there's direct anti-tumour activity, both through phagocytosis, but also through the production of TNF. And then we also have great data showing cross-presentation, activation of T cells, NK cells, and B cells. And so we are in a great place. We have dosed our first patient. I'm happy to talk about the design of the clinical trial, and we will hopefully read out the dose escalation data around Q3 of next year. So in the last few minutes here, I just want to touch on how we innovate at Myeloid. Andy Grove, founder of Intel, is someone that I look up to, and I keep telling the folks in the company, our scientists, that each product has to be 10 times better than the next one. And my job as a CEO is to have a balance sheet that allows us to get these iterative products into the clinic. And I think what I'll show you in a second, really shows that, the team is doing that. First and foremost, we've been able to take this protein biology approach and build specific CARs for specific immune cells. Now, one of the things that's really interesting, if you inject a non-targeted LNP, about 3% of the T cell compartment will take that up. Now, if you've developed T cells, CAR T cells, if you could show 3% of your circulating T cells were CAR T cells, you would feel very good about it because most of the time we use PCR. So we actually think that the current set of lipids that are out there for repeat dosing may actually be already sufficient to allow us to go into the clinic and target T cells and target NK cells. Secondly, mRNA has a long, long way to go before we've truly taken advantage of what it offers. I appreciate there are other people using circular RNA and so on and so forth, but using AI, we've been able to optimize sequences and structures, and this is mind-blowing. A couple of hairpin introductions here or there will result in extra expression for days. We've actually found microRNA sequences that essentially function as stabilizers of RNA, and we can actually get now up to 8 days expression of a straight mRNA construct within human cells. And finally, I've got 40 seconds. I really did want to touch base on this because I think this is really important. One of the barriers for delivering genes is the need to use viruses and have DNA templates. Retrotransposons open the door for us to be able to, on one construct that theoretically could be 14 kb in length, 1/4, right? 3 to 4 times larger than your viral vectors we use today, and do it. And we've actually been able to do this now by taking line 1 element. We essentially engineer the front end of it with a Cas9, and we've been able to stably deliver this into a site-specific and get up to actually now 2% transduction in cells. And so, I've run out of time, and I always do, because I'm so excited by what we're doing. I think we're, as Myeloid Therapeutics, trying to really push the envelope, and our focus is on curing cancer through building great science. And hopefully, in my 15 minutes, I've shown you just how versatile our platform is and how disruptive what we're doing can be. I think the future is a world that has allogeneic and in vivo approaches as well as autologous approaches, and I think as an industry we need to think about how we deploy these technologies carefully. But the myeloid cell compartment has something that T cells don't have, and that is the ability to traffic and penetrate and survive in the tumor microenvironment and the armory to activate T cells. We have many, many product designs we can go after, and we've proven, the team's proven, we're a company of around 40 people. And so we've actually developed everything in-house from scratch beginning March of 2019. And obviously our next milestones are actually proving that this stuff works in humans, but we're well on the path to do that. So, I thank you for the opportunity to present here, and we'll take questions outside just because of time, I suppose. So thank you.